So good morning or afternoon, depending where you are. Thank you all for joining us today for what promises to be a highly informative and impactful session. We're thrilled to have two distinguished speakers with us, each of whom will present cutting-edge research on the influence of circadian rhythms on health and cancer therapies. Our first presentation is titled The Influence of Chronodisrupting Events on the Metabolism of Environmental Carcinogens, an exposomic approach by Professor Konstantinos Makris from the Cyprus International Institute for Environmental and Public Health, School of Health and Sciences at Cyprus University of Technology. In this session, Professor Makris will share recent findings on how chronodisruptive events such as night shift work and irregular eating patterns affect the metabolism of environmental carcinogens like pesticides and benzene in humans. His presentation will also highlight the human exposome concepts and the exposomic tools used to comprehensively assess biodynamic exposures over time and space. We'll hear insights from several randomized controlled trials and panel studies that explore these relationships. Following Professor Makris, we're honored to have Professor Francis Levy, who is affiliated with both Paris-Saclay University in Villejuif, France, and the Department of Statistics at Warwick University, Coventry, UK. His presentation, titled Advancing Cancer Chronotherapy, will delve into the advancements in cancer chronotherapy, particularly how timing plays a critical role in precision and personalized cancer medicine. Professor Levy will discuss the latest clinical findings on chronomodulated chemotherapy and radiotherapy timing, showcasing how morning or evening administrations of certain treatments have dramatically impacted patient survival rates. He will also touch on the use of real-time circadian rhythm metrics and molecular clock technology in guiding treatment decisions and minimizing adverse events in cancer patients. Both presentations will provide invaluable insights into the future of circadian cancer medicine and its potential to revolutionize patient care. If you have any questions throughout the presentation, please submit them through the Q&A button at the bottom of your screen. We have allocated 15 minutes at the end of the presentation for questions and answers. Without further ado, please join me in welcoming our first speaker, Professor Konstantinos Makris.
Thank you, Amy, for the nice introduction. I will start my presentation by sharing my screen. I hope you can see it. Okay, so today I will give you a short introduction on the theme of connecting carcinogenic exposures from various environmental stressors together with the circadian and other metabolic clock influence on the disease pathogenesis, and how all this can be actually included in a relatively new methodological framework, the so-called human exposome, which tries to comprehensively characterize all environmental exposures that actually take place in a human's being lifetime or in specific time windows of susceptibility. So first, all the presentation results that I'm going to share with you would not be possible without the success and the collaborative spirit of my team, Dr. Harisiadis, Dr. Haibati, Mrs. Konstantinou, Mrs. Ioannou, Mr. Abibola, Mrs. Sursu, Mrs. Haddad, and Mrs. Kunde, together with excellent collaborators from ICANN School of Medicine and Mount Sinai, Professor Aurora and Professor Andra, together with Dr. Steneheim from Cancer Registry of Norway, Dr. Sana Rui from the Norway National Institute of Occupational Health, and last but not least, Dr. Alessandro Giuliani from the National Institute of Health in Rome, Italy. So the synopsis of my talk will basically center around with a short introduction on circadian clock functionality and the various applications of the term chronos. Chronos in Greek means time, in environmental health and biomedical sciences, and how perturbations due to desynchronized clocks could potentially affect environmental stressors like carcinogenic chemicals metabolism in the human body. So I will share with you some interesting chronometabolism, as we name them, case studies of these carcinogenic exposures and metabolism in human studies. So Some, maybe all of you are aware that a large proportion of our genes are basically regulated by the biological clock. And we know that very recently the Nobel Prize in Physiology of Medicine was actually awarded to scientists that discovered how specific clock genes are activated by specific proteins that allow them to degrade during the day and accumulate during the night. So they saw this diurnal variation, including the mechanism by which the light synchronizes the central clock in the human body. So it is very important to note here that from the moment that we wake up and the light hits our body, the circadian clock starts instigating and stimulating a series of physiological processes, such as, for example, the steep rise in blood pressure, the melatonin secretion overnight stops, and several other energy physiological parameters start to kick in during the day. And towards the end of the day, you have, for example, the highest blood pressure, and then towards the night you start having again the melatonin secretion until you sleep, and so forth. This is diurnal, let's say, periodic function in the human body regulates a series of functions like sleep, feeding behavior, hormone releases and transport, blood pressure, and body temperature. So these cues of the circadian clock, or synchronizers as they're called, such as, for example, day-night cycle, the meal times, other routines in our daily schedule, interact with the central clock in the suprachiasmatic nucleus, and together with the interactions with molecular clocks that govern the expression of specific clock genes, impact various physiological functions as we described above and earlier, such as physical activity, the body temperature, the cortisol or melatonin, and assuming that you have some kind of resilience and synchronization in the rhythmic function of that circadian clock, then a resilience and eufunctional of the circadian clock and the human body physiology exists. Otherwise, you have some kind of disruptions in the resilient cellular functions, leading to potentially to early stage markers of non-communicable diseases like DNA damage, oxidative stress and tissue damage, cell death, and we're gonna show later that these are basically key characteristics of carcinogens. So assuming that we have this so-called synchronization, which is a term that was invented by a Swiss glossologist, Dr. Saussure, refers to it as a totality of elements within a particular system that appear during the same time period. So the totality of these elements and the interaction of these systems elements within that network mention and comprise the synchronization pattern in the central clock, the peripheral clock, and the other molecular clocks that are spread throughout our body. And recent data showcase that moving from the once believed theorem of homeostatic biological constancy for various biological processes and functions, we might consider the so-called term homeostasis, according to Heraclitus, a Greek philosopher, that everything moves and everything is under constant dynamic processes. And this homeostasis approach actually has fueled recent, for example, discoveries such as the Nobel Prize finding in 2017, for example, or the Working Time Society consensus statement on how the circadian time structure effects on various xenobiotics, such as various environmental chemical stressors vulnerability, and how these circadian structure effects impact and differentiate the various homeostatic and compensatory coping mechanisms. To this extent, another modern concept has been coined by Professor Arora, a very good collaborator, about the biodynamic interfaces as an effort to better understand how humans interact with the environment they live in and the daily routines. And this basically talks about interfaces, and these interfaces integrates various biological levels and various biological systems that dictate the extent, the magnitude, the variance, and the degree of association and the timing between the various environmental stressors and health outcomes such as autism spectrum disorder, as we see on the right side of this slide, where we can see with different studies in Sweden, US, UK, various new system-wide biomarkers, which maybe are not familiar: determinism, entropy, mean diagonal length. These are novel biomarkers, allow me to say, of biological processes, early stage biomarkers of the autism spectrum disorder disease process. And another interesting finding that has also been recently observed is, if you see to the left, we see basically the normal, more or less, distribution of serum zinc levels in the US population of several thousands of Americans that a blood sample was collected, and you see how the majority of these values fall within the range of 60 to 120 microgram per deciliter. And if you move to the right, you have a graph that also shows the same exact range, but this time collected with the help of a few subjects. So we move from the scale of thousands of subjects to a few subjects that basically this time now they collected various blood samples within the day, within actually the span of 2 days. And you see that the variation, you have the lowest, let's say, levels towards the night for plasma zinc of about 60 microgram per deciliter, and the highest levels of them, you observe them in the morning. So you have like a similar variation of environmental pollutants in between and within subject. This is another interesting observation. And it is also important to note here, since we talk about the metabolism of various environmental stressors and how they interact with the disease pathogenesis, that in addition to the central clock functionality, we also have a series of peripheral clocks, such as, for example, the hepatic clock, which is a main system, peripheral system that governs the metabolism of all of these xenobiotic, all these external stressors that are absorbed by the human body, and they can be, actually, they are synchronized to cues such as food intake or even the air temperature cycles. It is also important to note from a series of animal models, and some of them human studies that actually have taken place with actual drugs, human drugs, that there is some circadian variation for most of phase 1 enzymes CYP450s. So this has actually been demonstrated with mouse livers that show peak expression patterns during the dark, which is basically the active phase for the animals. Similar patterns have also been shared with phase 2 conjugation of xenobiotics, which appear also to be under the influence of circadian rhythm, where you observe more glutathione conjugation in the early light phase, glucuronidation in the late light phase, and sulfation, another conjugation element, in the early dark phase. And these particular aspects, this particular circadian clock functionality that lead to synchronization or not, can, as we showed before, oftentimes has been associated with various metabolic and genotoxicity perturbations, such as DNA, let's say, toxicity, epigenetic alterations, the induction of oxidative stress, induction of chronic inflammation. So these 10 characteristics are basically IARC denoted carcinogen agent characteristics. So it would be interesting to note here that some of these biological processes and end products could be instigated with a chronic disease process and could be associated with external stressor and circadian clock functionality. So there are some chrono-disrupting routine events behaviors that could potentially move from a synchronization profile to a desynchronization and tissue damage profile or an inflammatory profile. So what are some of these events or behaviors? Night shift work. This non-standard work schedules is a very good example that has actually been charged already by IARC as a probable carcinogen, since we're speaking before about carcinogen agent characteristics. And night shift work has been recently coined by IARC that it is now a probable carcinogen. Another chrono-disrupting routine is for those that have to travel a lot and they have to change time zones in the travel. So this repeated consecutive travel across time zones could be another potentially chrono-disrupting routine that, of course, we know less about the influence on the disease process. Similar could be also with the story with various diurnally repeated behaviors, various 24-hour or so rhythms that may be circadian or not, such as, for example, some stereotypic activities like people like to drink coffee in the morning, or the various nutritional habits that may fall within a resilient and synchronized circadian clock functionality system or not. And of course, in addition to the 24-hour repeated behaviors, we might also have to consider some critical time windows, some critical periods in people's lives, such as periconception, pregnancy, childhood, that we consider are very, very important time windows for studying environmental stressor effects on human health. And some applications of the Chronos concept in studies, Professor Levy right after will share the pioneering studies that he has performed on chronotherapy and how the time-controlled drug administration, as you can see on the right side with the graphs, can actually lead to some beneficial effects, for example, improved sensory neuropathy and some other beneficial outcomes, or reduction in various side effects that might be typically present in cancer chemotherapy. Another example, the famous MAPEC chronotherapy trial with more than 2,000 participants. This is a randomized controlled trial, as you can see in the bottom right side of the graph, when people switched from actually taking all three medications in the morning by switching one of them being taken at night. You can see in the bottom right side of this graph, you can see a very clear separation and the reduction in the systolic blood pressure levels of the individuals compared with those that took all of the three medications in the morning. Another interesting result that relates to the Chronos application and the concept has to do, it's coming actually, now we move from the randomized controlled trials to observational studies. This is an interesting cohort of patients that have been admitted to hospital for self-poisoning, either with pesticides or oleander plant. And the study, this observational study, showed that the human mortality after the poisoning was in part determined by the time of poison ingestion. So, for example, with this graph on the right, you can see that the diurnal intestine absorption, either pesticides or oleander, was explained by the fact there is increased peak glycoprotein activity at night, which means reduced absorption, and therefore that would decrease the fatality from oleander ingestion. The oleander data is actually the black line, as you can see here. So at night we have reduced lower probability of death, contrary to what you observe with the organophosphorus pesticides, because at night it has been observed you have a higher CYP450 activity in the evening, which means you are increasing the metabolic activation of these parent pesticide compounds and therefore higher probability of death. Similar results with this time of administration have been also observed with human drugs, like the hypolipidemic simvastatin drug, where dosing at night, when compared with morning dosing in a randomized control trial, lowered significantly LDL cholesterol levels. And all these Kronos applications fit very nicely with recent policies and directives at both sides of the Atlantic Ocean, the personalized prevention schemes that fall within the personalized medicine scheme that defines personalized prevention, the tailored medical models to the right person or risk subgroups, and important to mention here that explicitly mentions the fact that they have to be delivered at the right time. And this is where what we want to emphasize. And personalized or precision health, personalized medicine or precision health research programs such as the All of Us of NIH in the US and the European Council in Europe conclusions that they put forward the legal framework for personalized medicine initiatives in Europe actually provide the policy framework for various Kronos application. For example, a very nice example by Professor Baccarelli, where they talk about how precision medicine can be also incorporated into environmental health and to primary, secondary, and tertiary prevention schemes. Another important and last application I would like to bring forward is, as you can see here to the right, the application of chrononutrition. And there is the very important low-hanging fruit from many chrononutrition studies that eating late is not good for human health. And this should be emphasized more and more in policy fora because it has been shown, particularly by Professor Panda's group from Salk Institute. You see on the right that how time-restricted eating, which actually is defined as eating within a smaller, with green color, daily window of eating, compared to the left side, more broad window of eating. How this time-restricted eating has been shown that reduces body weight, glucose intolerance, hypertension, etc. So having in mind these applications, these Kronos applications, particularly nowadays, it is very important and take into account, of course, the circadian clock functionality to bring forward a more comprehensive and holistic methodological framework such as that of the exposome, and how, for example, we can actually adapt and adjust the human exposome main 3 exposomic domains that extend from the internal exposome as onion layers outside the human body, covering a suite of different stressors that impact physiology and human well-being and quality of life. And if you see to the right, for example, we bring in concepts like circadian clock rhythmicity, oxidative stress inflammatory biomarkers as important internal exposome characteristics, specific external characteristics like, for example, shift work and other chrono-related routine behaviors or lifestyle that people usually follow either at work or at their leisure time. And this somehow combined with various policies of the industry and regulations about shift work, the various job characteristics, etc. If you combine all these different exposomic domains across time and space, you would have a much better characterization of the disease profile. And here we try to bring together how the external exposome time-stamped activities that are potentially chronodisruptive or not. It's important to link them and study them at the interface with the functionality of both circadian and metabolic clock systems as part of the internal exposome in humans, and how then this characterization of the metabolic, circadian and other biological processes as intermediate, for example, biological layers of information could facilitate linkages by studying the interface between various end points of disease that in certain cases will lead to synchrony, therefore resilience, or in other cases where this desynchrony could lead to differentiated toxicity risk. And these are some examples that we also believe the better characterization of critical time windows and the health trajectories could potentially lead to improved understanding of the disease process. And to this extent requires a lot of repeated measures in time. So we need to better understand the temporal evolution of both exposures and outcomes in human studies. In order to do this, it's not easy, it's not practical, it's very expensive oftentimes. Nowadays, we might be having more technological advances like sensors, for example. And with sensors, you can get a lot of data in 1 minute intervals or not, for example, where you get this individual level skin temperature gradients. And with this 1 minute interval skin temperature gradient within a day, you can see, for example, differences in this mosaic plot of the so-called recurrent quantification analysis that provide you with an alternative characterization of the temporal variation and temporal evolution as people, for example, live in an urban setting during summer, and how this mosaic, you see, is differentiated when people move to mountainous nearby, totally different climatological scenario. Another example is also the application of teeth, and Professor Arora actually with X-rays, they can, just like the layers of trees, they can capture the temporal exposure of a child, and they use this very high in number of repeated measures to visualize certain new network-based biomarkers, such as entropy, such as determinism, to characterize and discriminate children from ASD status to a non-ASD status. These are very interesting new technological advances that could potentially allow us to move forward the improved temporal evolution dynamics. And of course, life course multi-omics temporal dynamics, that requires that we take samples, repeated blood samples or repeated urine samples. This is a very nice study by Professor Snyder's in California, where they were monitoring healthy middle-aged individuals from 40 years of age, as you can see on the x-axis, all the way to 65, and they used a suite of different multi-omics capabilities and platforms from transcriptomics, proteomics, metabolomics, cytokine, clinical laboratory testings, and you see very clearly that there are two unique inflection points in this non-linear temporal dynamics of these healthy individuals. So there are these two inflation time points around 44 and 61 years of age. It appears that these are some critical time windows of susceptibility, that something happens with respect to aging. So these are prime examples of how Chronos global dimension can be actually incorporated into study designs of various diseases. And I'm going to close with some quick examples of chronometabolism of carcinogens with some non-pharmacological health intervention studies we have conducted, or panel studies. This case study number 1 actually studied how the cleaning activities with chlorine, regular chlorine, how we conduct cleaning activities at home with chlorine disinfectants, and whether the time of the day that cleaning takes place could have an effect on the metabolism of the disinfectants. And we looked in particular in urine, we looked into the trihalomethanes, which is a disinfection byproduct. So what we see here in these circles, we see in gray, these were urine samples that were collected before the cleaning activity, and with white color, these boxes are collected right after. And you can see how during the daylight, the median values decrease, but when you conduct the cleaning activities at night, you can see what you expect. You can see that you have an increase in the levels of disinfectants, disinfection by product exposure, as you can see here, pre and post cleaning activity. Another chronometabolism study, this is coming from a controlled trial with healthy individuals where we ask them to basically consume fruits and vegetables, conventional fruits and vegetables, either in the evening time or another week to consume them only during the morning. So assuming we actually gave them the same number, the same portions of fruits and vegetables, and you can see that when you compare the running day with green here, you see that when people consumed the fruits and vegetables at night, you can see what you expect usually to see, the increase in the body burden of the measured pesticides. However, when we asked people to consume the fruits and vegetables in the morning, we did not have basically a statistically significant difference in the levels of these pesticides compared to the running day and the rest of the days that the intervention took place. This is another example of the differentiated chronometabolism of pesticides that are found as food contaminants in fruits and vegetables. Last case study that I want to share with you is a BTEX, a benzene chronometabolism study that we did with gas station workers in Bulgaria. So this study designed here, they work 2 days for 12 hours in the day shift, then they have 2 days off, and then after 2 days they start working on the night shift. And by collecting urine samples of these individuals from the gas stations, as you can see here, we were able to show some interesting trends. So on top, you can see the benzene levels, how they vary, and on the right is one of the main validated metabolites of benzene, and in the bottom, the ethyl benzene patterns and the mandelic acid as a metabolite of ethyl benzene. Let's look for now only on urinary benzene levels, where you see the levels of urinary benzene pre-day shift and end of night shift. We did not see much difference, but you see when the workers were sampled after the night shift, lower levels of urinary benzene were detected in these workers. And nicely, this reduction in the urinary benzene levels was reflected in the benzene-specific metabolite that was measured in these individuals. So I'm closing with some take-home messages, more or less, and maybe some food for discussion and thought. I would like to start with a question that does sampling time in the day or characterizing the timing of the exposures influences the body burden of non-persistent xenobiotics? We showed that actually, yes, this is the case. And when you do take into account the timestamp of these exposures in the day, then you might be able to track and observe and chrono-differentiated body burden of metabolized xenobiotics, non-persistent xenobiotics. Secondly, does the exposure timing of environmental carcinogens matter in order to improve the assessment of the associated disease risk? Yes, and that's why we need better studies for that. And the Chronos dimensions and the interfacial interactions with circadian metabolic clocks might be very important to elucidate how the systems of time control activities, behaviors, and pollutant exposures could be implicated with a suite of biological endogenous response. So we need to study them both and study the interface of these two external stressors, timestamp external stressors, with the endogenous biological response in order to better characterize the downstream health outcome. And we believe that the precision health and the personalized medicine and the personalized prevention schemes that target multi-omics exposomic components that dynamically evolve in time would be very important to take into account how Chronos global dimensions could actually affect these dynamics. Last but not least, we believe that we need to have better designed observational and experimental studies, first of all, that are well powered. Secondly, that the attempt to include a high repeated measure sampling frequency of biospecimen and a relatively long monitoring of the intervention duration. Last, you need to incorporate multi-omics, exposomic tools in order to have some success advancing science in this new biology era that we live in. Thank you very much for your attention. I believe now we're gonna continue and we're gonna leave the room for presentations, room for questions after the presentation of Professor Levy.
Hello. Can you see my slide?
Yes.
Okay. So thank you very much for inviting me to speak here. One second. Okay. So my talk will complement that of Konstantinos and will focus on the medications, the toxicity and efficacy of the medications that are used to treat cancer according to circadian rhythms. And okay. So, as you now understand, we have a circadian timing system, which is composed of physiological rhythms in humans, like the night circadian rhythm in melatonin secretion with peak level around 2 a.m. at night, circadian rhythms in cortisol secretion by the adrenal gland with peak level in the early morning, around 8 a.m. in humans. Rhythm in core body temperature with high levels in the afternoon and trough at night, which is critical for sleep, for triggering the sleep at night. If the body temperature falls, does not occur at night, then sleep is very disturbed. Also circadian rhythm in heart rate, in psychomotor performance as well, which is better in the late afternoon, and obvious rhythm in rest and activity that can be monitored via actigraph devices. So these rhythms have to be considered as biomarkers of a complex circadian timing system organization, which involves the suprachiasmatic nuclei in the brain, molecular clocks that reside within each of our cells of all our organs, and their coordination by circadian physiology rhythms, especially core body temperature, cortisol, and rest activity, as well as sympathetic, parasympathetic tone rhythms. So this system, system basically attests to the fact that the circadian rhythms that we are observing are truly endogenous and are generated in the cells and in the whole body by the circadian timing system, which involves three components: the pacemaker in the brain, the physiological rhythms, and the molecular clocks in each of the cells. Now, the molecular clock has already been described by Konstantinos, And so I will pass this slide to move to what I will be speaking of. I will be speaking of the influence of the circadian clock on the success of immunotherapy of cancer first, then on endocrine therapy, then on cancer chemotherapy, and I will be discussing the impact of circadian disruption on cancer risk, treatment failure risk, and the recent developments that really allow us to track our circadian timing system in a non-invasive fashion so that we can foresee the personalization of circadian approaches to improve health and well-being. So I will start by the circadian immune system. Why? Because there have been recent data that highlight the relevance of timing for the efficacy of immunotherapy of cancer. As you know, immunotherapy of cancer has offered a new, a major therapeutic efficacy to combat many cancer types. My younger colleague, Dr. Karabwe, who is here, observed that when he was seeing his patients who were treated in the morning, they came several months later to see him in clinics, while those patients who were treated in the afternoon, he didn't see them after a few months. And so he wondered whether this could be due to the impact of timing of immunotherapy. And indeed, there is a large number of publications that support the idea of a circadian immune system. Circadian immune system, which in particular is revealed in healthy human by the fact that the naive CD8 plus cells that are really involved into the efficacy of this immunotherapeutic, of this new immunotherapeutic agents, and the neutrophil and effector CD8 plus T cells, which are also involved in the efficacy of these medications, really display prominent circadian rhythms in human beings. Now, what was found is the following in two publications that appeared only a few months apart. Is a first publication by a group in Atlanta, which has revealed that if you give immune checkpoint inhibitor, say immunotherapy, during the early time of day, say before 4:30 or after 4:30, this impacts significantly on the survival of the patients. And what Dr. Karabwe had shown even before this in a preliminary report, but also in a full publication afterwards, is the fact that the same was observed even more prominently in patients with non-small cell lung cancer with a 3.6 increase in the median survival of the patient treated predominantly in the morning as compared to those treated predominantly in the afternoon. So in order to investigate whether this was anecdotal or this could be was reproduced in other retrospective studies, we have performed a meta-analysis of 13 studies published over less than 2 years, arising from 9 countries and 4 continents and involving a total of 1,667 patients. And this involved different types of cancer: melanoma, lung cancer, kidney cancer, bladder cancer, esophagus cancer. All these studies concur to show that treating patients with immune checkpoint inhibitors predominantly in the morning or in the early time of day, say before 4 p.m. in all instances, was associated with improved overall survival, as well as improved progression-free survival, which is indicated by this hazard ratios of an earlier death, basically, which means that overall you can double survival if you give these medications early in the day. Okay. So the main mechanisms that work have been identified in experimental studies, especially conducted in mice, highlighting again that administering immune checkpoint inhibitor in the early span of activity was more effective because there was a circadian rhythm in the infiltration of target cells, namely TCD8 plus cells and dendritic cells in the tumors, which was highest in the early morning hours or early activity hours. So the circadian timing system is closely interacting with the immune system. There are reciprocal interactions. As you know, the suprachiasmatic nuclei is controlling the synthesis and release of the cortisol and catecholamines, which are coordinating and playing role in immune system function and circulation. The core body temperature is also influencing this immune system, and in turn, the cytokine production by the immune system is feeding back on the suprachiasmatic nuclei. So there are complex interactions between the two systems at many levels of interventions. Now, the new immunotherapeutic agents work primarily by interfering with the PD-1 protein, which is at the surface of the CD8 effector cells, and or PDL1, which is on cancer cells, but also on myeloid cells. So these interactions appear to be profoundly controlled by the molecular clock. And to complete the story, today there are 26 retrospective studies that have shown what I have just shown you. Early time of the administration is better than late time of the administration. There are 3 randomized trials that are going on to further prospectively confirm these findings. Endocrine therapy is also interesting from my viewpoint because you know it's one of the main treatments of breast cancer, hormonal dependent breast cancer. My colleague, Dr. Giacchetti, has investigating the relevance of timing endocrine therapy within as an optional study that was part of a randomized trial testing the addition of everolimus to endocrine therapy as adjuvant treatment for breast cancer. And so overall, 325 patients were given tamoxifen, 530 were given aromatase inhibitors, and the patients were asked to choose when they want to treat to get these medications. And so overall, 511 chose to take it in the morning or in the afternoon, and 344 chose to take it in the evening or at night. So here we examine together and separately the impact of timing on the efficacy of endocrine therapy. The first finding when we examine separately is that indeed the ingestion of tamoxifen in the evening or at night time was associated with increased disease-free survival. This is more than, here you have 5 years, and here you are beyond 5 years. So it's a markedly significant effect of the timing of tamoxifen. It is the first time that this is shown. Recommendation for morning, afternoon, rather than evening, night. Sorry, recommendation for evening intake rather than morning, afternoon intake of tamoxifen. In contrast, however, you see that aromatase inhibitor, whose mechanism of action is completely different from tamoxifen, does not seem to play, that timing of aromatase inhibitor does not seem to influence outcome in the patients. Interestingly also is the fact that the time dependency of tamoxifen effect on disease-free survival is even prominent in premenopausal women and much more damped in postmenopausal women, calling then for further investigation of the circadian rhythms, the modifications in the postmenopausal women as compared to the premenopausal women. A possible explanation of this improved efficacy of evening intake of tamoxifen might be related to the recent publication by a Swiss group of Aceto that indeed patients with metastatic breast cancer had cancer cells that circulated more frequently at night as compared to daytime. Not only more frequently, so this is all the individual patients. In red, you have when it is more frequent at night, so it's almost all patients. But it is not only a higher number of circulating tumor cells in the blood, but it is also that these circulating tumor cells are a lot more aggressive in terms of metastatic potential as compared to those few which are detected at daytime. So it's very likely that if you give some medications at the end of the day, you are more effective on the circulating tumor cells that could give rise to metastasis or relapse of the cancer cell. A third aspect I want to highlight is the cancer chemotherapy. We were the first to publish in the first randomized trial on this issue. But today, 18 randomized trials have been reported in a large number of patients. This systematic review by a group from the Netherlands has highlighted that in 77% of the trials, there was a benefit for chronomodulated chemotherapy adapted to circadian rhythms regarding toxicity, and that in 3 of these trials, there was also an improved in efficacy, and in the 11 other trials, there was efficacy was maintained. In no trial, there was a decrease in efficacy. So they advocated for the further administration of chronomodulated chemotherapy because there is at least maintained efficacy. But of course, the main message is that we need more and larger randomized controlled trials to provide recommendations for clinical practice and also to investigate chronotherapy with other cancer modalities like immunotherapy or endocrine therapy that I just showed you, and also to investigate striking sex specificities. So I will move further. Now regarding circadian disruption. So when we speak of the chronoexposome, mainly the alternation of light and darkness, but also temperature cycle, noise, social interactions, daily life, activity, time of food, time of ingestion of medications. All this is contributing to impose on our body good circadian rhythms. If good circadian rhythms are working, are effective, then usually we are in good health. So good circadian rhythms have been associated with the well-being and a better health. Now, if these synchronizers are disturbed, or if their perception by the body is disturbed, then the molecular clocks, then our suprachiasmatic clock does not know what time it is. Our molecular clocks can indicate different times. They are not coordinated anymore. And then there is a risk of pathology. The risk of pathology that have been shown both in epidemiological studies and in experimental studies are the development or worsening of cancer, cardiovascular diseases, GI diseases, liver and metabolic diseases, neurodegenerative diseases, and infections. So this is a nice example of the relation between circadian alterations and the outcome of subjects. So this is a large study that involved nearly 100,000 patients, people, sorry, and that wore an actigraph when the rhythm was marked. Is the highest. They divided it into 5 quintiles. So this is the highest quintiles with large amplitude rhythms in Western activity over 7 days. This is the lowest quintile where the rhythms are markedly damped. And what is the main findings that are striking is that there was a follow-up for over 6 years of all these people. And those who had a disrupted rhythm in activity developed more cancers and other diseases within the 6.5 years that followed. And those who developed cancer, cardiovascular diseases, or respiratory diseases died more than the other ones. So I think it's an interesting study that is also indicated in the shift work that was discussed before. Here, because night shift work has been associated with the development of cancer, we have tested the relevance of telemonitoring of circadian rhythms in rest activity and chest surface temperature for 1 week together with classical epidemiological measures. So the nurses were carrying this sensor, which provided information on their activity and temperature every minute. This is the kind of pattern we got in day shifters with a very regular rest activity rhythm, very regular chest surface temperature rhythm, detection of a prominent period in chest temperature, and marked alternation of rest, intermediate activity in pink and high activity in red, which is using the hidden Markov model. The 77 day shifters had quite reproducible individual patterns in their rest activity rhythm. And 70% of them had a significant rhythm in the chest surface temperature. In contrast, this is an example of the night shifter with a prominent disruption of the rest activity pattern, a prominent disruption of the temperature with no detected 24-hour rhythm. And what you see among these 66 night shifters is that there is a prominent heterogeneity in their rest activity rhythm, and only less than half of them had a significant temperature rhythm. But the most interesting finding is that the alteration of critical circadian rhythm parameters was significantly predicted by the number of years exposed to night work. So it means that you have a real chronoexposome effect on the deterioration of the circadian timing system. And if this system is deteriorated, it is possible that this contributes to the development of cardiovascular and malignant diseases, especially breast cancer in particular. So to conclude this, we are currently integrating this approach to detect the risk of developing emergency hospitalization in patients with pancreatic cancer. So we are telemonitoring the rest activity temperature rhythm together with symptoms and body weight with data that are automatically analyzed into a platform here with feedback on information on circadian parameters and alerts to the team. The study had just completed accrual of 59 patients, so we will know whether this approach is bringing out significant benefit for the patient's quality of life and response to treatment. So to conclude, I would say that we now have tools that can be integrated into a digital platform to address the issue of personalized chronotherapy. Why? You have tall people and small people. You have heavy people and light people. You also have you also have, night owls and early larks. And you have all the intermediate patterns that exist. And we have learned that you need, in order to get the most out of circadian rhythms for the treatment and the risk detection, you need to have a personalized approach. It's already good to have a group approach, but clearly, for instance, women do not respond the same as males to the same chronotherapy schedule. Uh, uh, the risk, the improvement of efficacy of tamoxifen by evening administration is prominent in premenopausal women, but is dumped in postmenopausal women. We need to understand all this, and to do this, we need to be able to track how the circadian timing system and other systems are working in the individual people. We now are fortunate to have the tools, not only the tools for the telemonitoring of circadian physiology, but also the artificial intelligence tools that enable us to know whether the molecular clock of a given tissue from a single biopsy is working or not, and what time is it in this tissue. So we can combine all these approaches to foresee now personalized circadian medicine, both looking at risk and looking at treatment effects. I will conclude to say that the circadian timing system controls health and well-being. That means that we can use it to better prevent diseases, in particular cancer. The circadian timing of cancer treatment is an important determinant of patient outcomes for symptoms, quality of life, tumor responses, and survival. And that I didn't speak on this, but all this could be developed because of cellular, animal, and mathematical models of chronotherapy, which provide mechanistic insights into molecular clock dependencies and biomarkers. And finally, we must consider that there are sometimes very important between patient differences in circadian rhythms. that make it necessary to develop personalization of chronotherapy. Unfortunately, we now have in hands non-invasive metrics of circadian biomarkers and molecular clocks and chrono artificial intelligence, which make it possible to consider the development of personalized chronotherapies integrating host and tumor clocks metrics. Thank you for your attention. I want to emphasize that all this needs to be further validated in randomized controlled trials, and that we believe that chronotherapy should become part of new drug development strategies. I want to thank my collaborators, without whom what I showed you would not have existed. Thank you very much.
Thank you everyone for attending today's webinar, and thank you for staying a few extra minutes till the end. And thank you once again to our speakers, Professor Konstantinos Makris and Professor Francis Levy, for their valuable contributions. As well, thank you to all of you for your engagement and participation. We hope you have a great rest of your day.
Shall we allow for maybe a couple of questions to see if anybody wants to ask something? I don't know if anybody from the audience wants to ask a question.
I'm not seeing any questions come through yet, but if anyone would like to ask a question, please use the Q&A feature at the bottom of your screen.
Maybe I can ask a quick question before somebody writes something to Professor Levy. Thank you very much for the very interesting presentation, Professor Levy, and the complementarities. I see a lot of complementarities between our two talks. And what I want to ask you is, I was very intrigued by the result you showed, the differences between tamoxifen and the aromatase inhibitor treatments. And you said that there is the different results of these two treatments are due to some different biological action, different biological processes that govern these treatments. Do you want to elaborate a little bit more on that, those differences?
Well, tamoxifen is targeting the estrogen receptors at the level which are expressed at the level of the tumor cells. So if you have a tumor cell with estrogen receptor, tamoxifen will target the estrogen receptor on the tumor cells. In contrast, what aromatase inhibitors do is that they completely suppress any estrogen secretion. And by suppressing estrogen secretion, they will make estrogen receptors ineffective because they will not be stimulated by the estrogen. So they are completely different mechanisms of action. In one case, basically, you can target the receptor on the tumor cells. In the other, you make these receptors inactive by suppressing the endogenous hormonal secretions. Now, does this necessarily imply that there is no time effect for aromatase inhibitors? Quite frankly, there is strictly no preclinical study on these topics. There is no clinical study on these topics. These results are the first worldwide that have addressed the issue of the timing of endocrine therapy, whether it is in adjuvant or metastatic setting. These are the first data worldwide on the timing of endocrine therapy in breast cancer patients. So we need many more to complete the picture.
Yes, indeed. Thank you. Thank you very much.
I have a question for you because I'm intrigued by paracetamol liver toxicity. And you spoke mostly of the environmental toxicants. But what about the medications that, with epidemiologic or pharmacoepidemiologic studies, we are discovering adverse events that were not suspected. I think of paracetamol because it has a well-known circadian dependency in animal models, but in humans, there can be a marked liver insufficiency and cholangitis. And I was wondering whether there is any way to address the issue of paracetamol timing in humans. I don't know. I'm naive in this respect, but I was wondering after I heard your talk.
What you're hypothesizing is maybe correct. I mean, we haven't done these studies, and I'm not aware of any human studies. I'm aware of several animal models on the topic that have actually shown various effects of this drug administration at unique time points of the day on pharmacodynamics and pharmacokinetics of these drugs. That would be something interesting to study, definitely.
Yeah, because the treatment is liver transplantation. The treatment of cholangitis due to it's a rare event, but when it occurs, the issue is die or transplant. So
that's
it could
correct.
be interesting, but again, these are methods that are beyond our own expertise. So it would be interesting to see how this could be addressed at a population level, basically.
We can discuss further, definitely.
Yeah. Well.
Thank you. Thank you very much, and thank everybody. And we'd like to thank also Mrs. Amy for her assistance.
Okay.
There she is.
Thank you. I think there is no more question apparently.
Thank you. Thank you very much.
So no question. Everybody is happy.
Thank you,
Yes.
everyone.
Thank
Bye.
you. Bye.
Bye.